And he's actually a PhD. He was a PhD student actually in Glass Lab, and now he's currently a postdoc fellow in the lab of Adam Cohen at Howard University, where he explores how to use voltage imaging to learn new things about the spinal cord. Previously, he studied biology in Framework and Singapore, and has been working with zebrafish since his master's. Yeah, for the PhD, as I just said, he was in Claire's lab working on the role of mechanosensing neurons in the spinal cord. Thank you. First, floor is yours.
All right, thank you. Thanks for the introduction and thanks for having me. So yeah, let's dive right in. So I'm Urs. I'm a postdoc in Adam Cohen's lab, and the lab in general is, well, one of the things we're looking at is voltage or bioelectricity in all kinds of systems. And I joined with the idea to use zebrafish as a model to look at, to develop voltage sensing, voltage imaging, and look what we can do in terms of looking at voltage in zebrafish neurons. So I'm gonna give a bit of an introduction about sort of my view of voltage imaging in general, just so you get an idea about what sensors there are out there and what the challenges are before I go into my own work in zebrafish spinal cord. Okay, so why do we want to do voltage imaging in the first place? I guess some of the reasons might be rather straightforward. Change in membrane potential in a neuron is sort of a fundamental unit of information in the nervous system, and we'd obviously, if we want to understand a brain, we want to look at these changes in membrane potential, notably action potentials, and we can do that with very high fidelity in great detail using patch clamp measurements. So poke into a cell and measure the membrane across the measure voltage across the membrane. This is a sort of an invasive approach, and it's not really scalable. You can maybe record from a few cells at the same time if you're really good at it, but that's where it usually ends. So the other widely used approach, obviously, if you want to you look at many neurons in parallel, is use calcium imaging, express a genetically encoded calcium indicator in neurons, and like that you can look at thousands or tens of thousands of neurons in parallel potentially. Calcium indicators are report on a sort of a proxy of neuronal activity. Basically, they report on the increase in calcium that is due to the firing of an action potential. They're inherently slow, partly the kinetics of the sensor itself, partly the kinetics of calcium changes in a neuron as well. And there's just laser pointer. So this is just a comparison of the sort of most recent calcium sensors and in response to a couple of action potentials fired at around 80 hertz. And what you can see, and you might know that, is that while they report very nicely on the activity, you can't really resolve any individual action potentials. There are ways, but it's not straightforward. And then there's relatively slow kinetics. So calcium sensors generally more report on a low pass filtered version of the overall firing rate of a neuron. So clearly there is there's an area where you might want to use a genetically encoded voltage sensor to directly look at the cell membrane itself. And notably, if you want to look at more neuron than one at the same time, if you want to be non-invasive. And then there's also a thing that aren't really accessible with either calcium imaging or patch clamp. If you think about subthreshold voltage changes, for example, in many neurons in parallel, it's known that in some areas, the firing of neurons can be quite stochastic and uncorrelated. Threshold voltage fluctuations are much more correlated, and using a imaging approach of voltage of many neurons in parallel, you could potentially look at that. And then not going to many neurons, but zooming in into one neuron in particular, you can imagine that you can look at the voltage changes in different compartments of a neuron, say how it changes in one dendritic branch versus another, and how the voltage changes when the action potential races down the axon or something like that. So there's sort of many areas where an optical sensor of membrane voltage might be helpful. So how do we go about to have something like that? So in a neuron, you probably know this, at resting potential, it's hyperpolarized relative to the extracellular medium. There's a voltage gradient across the membrane. If we do patch clamp recording, we stick an electrode, we open a hole basically in the membrane, get electrical access to the inside of the neuron, and measure the voltage drop relative to the outside of the neuron. So the point I'm making here is that the voltage drop that you're trying to measure is across the membrane. So you need a protein that sits in the membrane if you want to have a genetically encoded voltage sensor. Proteins that are not sitting in the membrane will not experience the voltage drop, and thus will not report on any changes in the membrane voltage. And not only do they not report on the change in membrane voltage, but they actually contribute actively to the background noise. So a protein that's sitting somewhere in the cytoplasm and is fluorescing adds basically noise to your measurement. And a lot of the improvements of the most recent versions of voltage sensors didn't come so much from increasing sensitivity or brightness. That was one aspect, but another very important aspect was actually improving membrane trafficking to make sure that every protein that is produced in the cell ends up in the membrane and thus contributing to signal and not to noise. Okay, so we want a protein that sits in the membrane. It needs to report fluorescence somehow, so we can actually see it. And then this fluorescence obviously needs to change whenever the membrane voltage changes due to an action potential or something like that. So that's sort of the basic idea of a genetically encoded voltage sensor. And there's been development in this field for quite a while. And most of it was sort of proof of principle experiments. And recently, I would say that over the last couple of years, there have been a bunch of sensors that were developed that are good enough to be used in an actual animal to do actual neuroscience with them instead of just showing that in principle they can work. So I think, so over the last couple of years, they became to a level where you could reasonably say, okay, I have an actual neuroscience question that needs voltage imaging, and I'm going to use one of these sensors and actually try to answer that. And sort of in my view, there's sort of 3 sensor types these days that are usable in such a way, and I'm just going to talk a bit of the pros and cons of each of them. The one that I'm using for all of the data I'm going to show later is called Archon. It was originally developed in our lab and then took a detour, got improved in another lab. And the basic principle is that it's a rhodopsin that's in the membrane, and originally it's a proton pump called Arch, and the pump function has been mutated out, and the protein itself is weakly fluorescent, and whenever it gets protonated or deprotonated, depending on the membrane voltage, this induces a conformational change that changes its fluorescence, and that we can measure that. The advantages of this one is that it's excited relatively in the red spectrum of things. So it's typically excited with like 630 something nanometer light, and then emits even further red, which makes it makes it relatively straightforward to be combined with optogenetics. If you want to not only measure the membrane voltage, but also manipulate it, you can express a light-gated ion channel basically in the neurons, and to be able to independently measure and manipulate the neuron, they need to be spectrally separated. So you need to use a different light for the imaging than you use for the manipulation. And since this Archon sensor is relatively red, it's easily combined with sort of the current optogenetic tools that are out there. It has a relatively large change in fluorescence upon change for any given change in membrane voltage. The downside is it's quite weakly fluorescent. So you need a lot of light to get a decent signal out. A second
I got a
sensor that was developed recently is called Voltron. Yes. Yes.
I was just wondering, where is the Archon initially from? Is it like from what species, I guess? Because often they take them from bacteria or something, no? Like, is this like a protein that exists like this in nature? I mean,
yeah, it's a proton pump, I believe, from some algae.
Okay.
I forgot the exact species, but yes, it's usually a proton pump that basically uses light as an energy source to pump proteins, and the way it's working here is sort of in reverse. Instead of using the light and pumping protons, the pump function is gone, and the change in protonation or deprotonation changes its absorption spectrum and thus its fluorescence.
Okay, yeah, that's very cool.
And that's how you can report it.
It's cool to see where it comes from. Okay,
yes.
Thank you.
The Voltron sensor is based on a similar type of protein. It's not ARCH, it's another proton pump. I think it's called ACE. Doesn't really matter, but it's the same principle. It's a rhodopsin that sits in the membrane, but instead of looking at the fluorescence of the protein itself, it actually looks at the fluorescence of a fluorescent tether. And this tether is close enough to the protein that it can undergo Fred energy transfer. And that Fred efficiency is basically dependent on the exact conformation of the membrane protein. And thus, whenever the membrane voltage changes, the protein conformation changes, and then the Fred efficiency changes. So what we're looking at is the signal of this fluorescent tether that undergoes Fred with the membrane protein. In this case, it's not actually a fluorescent protein that is tethered, but a protein that can capture a dye. So whenever you use this sensor, you express it with whatever genetic expression system that you want to use, and then you just add the dye. In a zebrafish, you can just actually add it to the water for an hour or two, and it's soluble enough that it enters the neurons and binds to this this protein here, and that's what we're actually looking at. The advantage of that is that it can be very bright because you can use a very bright dye, and potentially more photostable. The change in fluorescence upon any given change in membrane voltage is generally a bit smaller. But since it's so bright, for all, it depends a bit. But I've tried both of them, and there is not a fundamental difference in basically signal to noise that you get with Archon or Voltron. One might be a bit brighter in some circumstances, but it's less different than you might think, because Voltron has a relatively small change in fluorescence. Archon has a big one, but Archon is very weak and Voltron is very bright. So these two sort of average out roughly anyways. Yeah, this one is potentially a bit easier to use with, because you generally need a bit less light, and it's used with green to yellow, depending on what dye you're actually using, wavelengths, which is sort of more standard colors on a microscope. It's not as straightforward to be used together with optogenetics, because yeah, it's used with green to yellow excitation, and that goes more into the spectrum of the current optogenetic tools. And what you have to keep in mind is that for all of these, you need quite a lot of light to see anything compared to what you usually would use for calcium imaging. So even if spectra are reasonably well separated from optogenetics, because you can use so much light, you might still, spuriously activate your optogenetic tool in a situation like here.
Can I ask around what range, like what's the scale that the light is required to activate?
In terms of power?
Yeah, power.
So for Archon, it's in the well, it depends on what measure you're using, but it's actually in the kilowatts per centimeter square. So it's really quite a lot. But for zebrafish, well, it's red light, and most of it actually goes through. So I haven't seen any real adverse effects of using so much high power. I mean, you bleach your sensor, obviously, but that's sort of expected.
You bleach your voltage sensor?
Yeah, I mean, the voltage sensor, bleaches over time as any fluorescent sensor does.
Over how long?
In this case, I think my recordings, the longest I've done is sort of on the range of a few minutes. The mouse people in our lab went up to maybe 5 or 10 minutes. The Voltron, so from the original Voltron publication, I've seen data that goes up to maybe 15 minutes or something like that. In general, you're in the minutes range, sort of. You're not going to do an experiment like for calcium imaging where you.
Yeah, and the signal gets significantly
and the signal goes
dimmer.
Exactly, becomes dimmer and dimmer until at some point you just you lose it basically. And this is all relative, right? I mean, if you want very high fidelity signal, you have to use more light and then it bleaches faster. If you have a neuron that expresses very, very brightly, you can use less light and image for much longer. If you just want to, if you have spikes in your neurons that are relatively long lasting, you can image at lower frequencies and then use less light because you integrate more light over any given frame. So it depends a lot on your actual situation, but it's somewhere in the minutes to tens of minutes range.
So the little question. Oh, sorry. Go for it.
Oh no, I just wanted to ask if you so once you use it for 15 minutes, is it then never usable again? Like, is this like a one-time?
Yeah, then it's gone. It's bleached. I mean, you could potentially wait and for the neuron to produce new sensor, but that particular molecule in the cell is then gone.
So you would need a new organism, or you just go for a different cell? Like how?
Well, if you, I mean, you should design your experiment such that you can measure whatever you need to measure within this sort of time frame.
Okay,
but yeah, you can go to the next, another optical section or another region of the brain and keep on measuring. I mean, but this particular cell is sort of like, yeah.
Okay. Okay, cool. Thanks.
I wanted to ask you, so since you're doing experiments in mice and fish in parallel in the lab, did you observe smaller signal in the fish where often the action potentials are smaller amplitude? Did you see that, or it was?
Yeah, so there's all kinds of stuff that makes it seemingly harder to get good signal in fish, I think. One is that the cells are smaller, so you record from fewer protein, basically. The expression level also might be different because they are typically optimized for a mouse system and might not express as well in fish neurons. And then the action potentials themselves, sometimes they're quite fast. So in our lab, for example, the mouse people had great success in recording pyramidal cells, but it's very hard to get any decent signal from PV neurons in the hippocampus because their spikes are so fast that you need that. Yeah, often you can't record them, or they get low pass filtered and then become very small. And I think I have a similar situation in some of my spinal cord neurons that the spikes become very, very small. And it could be a combination of a fast spike that is low pass filtered with this by the sensor. In the imaging, it could also be that the cell intrinsically has a small spike, at least at the soma where I'm recording. So that I'm not exactly sure, but in some neurons, it's really hard to actually resolve spikes. Yeah. The third sensor that is sort of on the market is called ASAP, and that's based on a different system. It's also a voltage sensitive protein in the membrane, and that is tethered to a fluorescent protein. But instead of undergoing FRET like in Voltron, the conformational change in the membrane part of the protein pulls on the fluorescent protein and thus changes its fluorescence. And the work principle is sort of similar to the calcium sensors, where calcium binding changes the conformation of a fluorescent protein. So this one, what makes that sort of stand out from the other two is that it's the only one that is capable to be used with 2-photon microscopy. So the fluorescent part here is based on a GFP, I think, and depending on your experimental situation, you might want to go very deep into tissue and use 2-photon microscopy. And this is the only one that actually works with 2-photon. The other two don't, for reasons that are not exactly clear. And we can talk about it later if you're interested. But if you want to use 2-photon at this point, ASAP is the only one that is known to properly work. It has slightly slower off kinetics, so the spikes sort of are a little bit dragged out, but it's still perfectly usable to resolve single spikes. And the same with Voltron, because it's more in the green range. It's a bit less straightforward to be used with optogenetics. Okay, so in my opinion, these are the three that are sort of on the market, and in principle, you could, depending on your experiment, pick any of these and just go for it. Just want to put one slide about sort of the challenges compared to calcium imaging, which is a very established technique that is relatively easy to establish in any kind of lab. The voltage imaging suffers from two sort of main challenges. One is that compared to calcium imaging, we're now not measuring from a volume, so that's entire cytosol that has calcium indicator in a neuron, but we're from a surface, because all the protein that report on the membrane voltage can only sit in the membranes, so on the surface. Or if you think about it as a cross section through a cell instead of a disc, now you're measuring from a line. So the number of proteins that we're measuring from is much smaller compared to calcium imaging. Not only is it much smaller, because the events that we're interested in are often action potentials, and they happen on the timescale of milliseconds. So to resolve these, inherently we need to image very fast. Hundreds of frames per second, ideally, kilohertz imaging rates, to have the hope of resolving, the fast action potentials that are happening. And not only are we imaging from fewer protein, but we also need it to do it much faster. And both of these combined make it such that the sensor needs to be relatively bright, or speaking the other way around, on any given frame of our recording. What we're constantly fighting against is basically weak signal from our sensor. The main challenge is to get enough signal from a neuron per each individual frame to be able to record anything that is above basically our background noise. Because we need to image very fast, usually the easiest is to opt for a camera imaging readout, because you can go fast and parallel over many neurons. You can also use scanning approaches, but a normal sort of 2-photon or confocal scanning, if you go for more than one cell at a time, would be way too slow. So either you look at a single cell at a time, where you would just scan around and one cell that could be done fast enough, or you need some more advanced random access kind of scanning approaches that could potentially image from many neurons in parallel at the high rates that are required to resolve any of these events. So these are the main challenges, I think, in my opinion. And having said that, I just wanted to show you some sample traces that are now from my own data, because it does work. As I mentioned, these sensors are at the level now where we can use them in a living animal to record electric activity from neurons. And this is just an example. I'm expressing the Voltron sensor in a zebrafish spinal cord. We're looking at it from the top. So you see the left and right side on the top and the bottom of the image. And then these are just two traces from two neurons that are sitting there while the fish is spontaneously swimming around. I mean, it's sitting under the microscope and it's paralyzed, but it's trying to swim. And immediately you can see that there is some oscillations going on, and then there are some small spikes on top of these oscillations. And maybe this oscillation, one of the neurons suddenly doesn't spike anymore. And so you get very, very rich kind of data, as to be expected, as you would with a patch clamp recording, only that you can do it not only from a single cell or from a few cells, but potentially from many, many neurons in parallel. And
so also on this slide, did you inject the construct to have it at high level, or did you use transgenic animals?
This is a transgenic animal. Yes. Yeah. And I am going to stop sharing and share again and optimize for video. So you guys can actually, is the quality still decent, or is it?
It is.
Okay. How does this work? Sorry.
To me, it's all a bit blurry, but I don't know whether that's just me.
I think it'll get better in a
Okay.
second or two, hopefully.
Can you make it full screen, maybe? Because it's small.
The video?
Yeah. Could you?
Or the entire presentation?
The video. Could you make it bigger?
Yeah, I'm trying to actually have it run in the first place, which it doesn't seem to do. Okay, let's not get hung up here. It was just a little sample. I'll have other videos later. But basically, yeah, from the traces, you can see this activity. And as I mentioned, this sensor is relatively straightforward to be used with optogenetics. So here's an example trace where we express not only the sensor, but also
I think that you since you went for video optimization, we lost the resolution of the.
Yeah, I'm going to go back to.
Okay, cool. Sorry.
Normal. Is that better?
Good.
Okay. So yeah, so in this example, I'm expressing also an optogenic actuator in the same neuron, and if we give increasing pulses of blue light, in that case, we can make the cell spike. And this is not spontaneous activity. This is induced by sort of the experimenter in that case, and that's just to show that.
Sorry, to me it's still blurry. Is that yeah? Is it just me?
It's good for me.
Okay.
Too.
Okay, so it might just be me. Okay, sorry.
Okay, so what am I actually using these sensors for? So let me step back one second and talk about sort of my motivation for my sort of neuroscience work. And okay, these are all videos, but they're probably not gonna look great on the over Zoom, so I'm not going to run them. But basically, if you think about an animal moving around, actually, if you think about any kind of neuronal activity, ultimately, it usually leads to some sort of muscle activation. That's sort of the ultimate output of the nervous system. And a huge part of this muscle activity is related to movement, right? And the movement is generated in the spinal cord, and the core functionality of the spinal cord is generating an oscillation that regularly activates, you know, legs on the left and right side of the body, or wings, or have a tail wiggle back and forth. And this core oscillator, or central pattern generator, has been studied for quite a long time, and we have a relatively good understanding of how it is generally set up. At least there's on each side an excitatory pool of neurons, and via inhibitory neurons cross inhibits the other side, and this starts oscillating, goes back and forth, and then connects to motor neurons, activating the muscles. And while this sort of general idea is very established, you can immediately imagine that there has to be much more to the story. You can walk faster or slower, you can turn around, you can walk more strongly, for example, if you walk up a hill with the same sort of speed, and all these kind of modulations and changes in the general oscillatory activity are much less understood, and probably also differ quite significantly from animal to animal. And so what I wanted to do once I had the general voltage imaging working is to globally look at as many neurons as possible at the same time, while in my case, a lover zebrafish is doing a behavior and characterize what types of neuronal activity are actually visible in a spinal cord during an actual behavior. Most of these patch clamp recordings are, of course, done in ex vivo preparations in slices, or in case of zebrafish in a sort of a semi-slice where you at least open up the spinal cord and go in there with electrodes. And so using an imaging approach would allow us to have the fish in a situation where it can behave relatively naturalistically, and having a voltage imaging approach allows us to really look at the fine details of the activity that's going on. Even a slow sort of forward swim of a zebrafish larva is around 25 hertz, and that is too fast to resolve with calcium imaging. Anything more than is basically saying the fish is swimming or not swimming. If you want to look at any of the fine details of the oscillations, when exactly the spike's happening, and so on and so forth, you need basically voltage imaging. Okay, so the system I'm using to measure these measurements is a light sheet setup to illuminate only the neurons that are in the focal plane of my imaging arm of the microscope. I'm coming in from the side with the excitation light that is generated, comes in as a sheet, excites only the focal plane, the neurons in the focal plane. Then I'm imaging from top with the camera all the neurons that express the sensor in the focal plane. And like that, I can potentially record for many neurons in parallel. And I said I would want to measure a fish while it's sort of behaving as naturalistic as possible. Obviously, they are sitting under a microscope. They have to be paralyzed. Otherwise, they move and I can't really see anything. But what I'm doing here, and this is sort of, I didn't invent that, is recording with an electrode from the motor nerve bundle that innervates the muscle, an electrical signal that is basically a proxy of what the fish's intent to swim. And call that fictive swimming. And not only am I recording this, but I'm actually giving the fish visual feedback based on this signal. While it's sitting under the microscope, it sees a grating. And when I externally apply sort of a flow to this grating, fish try to start swimming because they want to stabilize their situation in the world and try to follow that grating. And by measuring its fictive swimming signal and feeding that back onto the speed of the grating, can basically move its scenery in the opposite direction. So it gets the impression of actually compensating for the optic flow that I'm imposing. And this is helpful in this case because I want the fish to swim as naturally as possible. Often, if you induce swimming with an optic grading under microscope and you don't have the feedback, at some point they give up or they start trying to swim very, very strongly. And I want them to swim as normal as possible. So I have this sort of closed loop virtual environment that the fish is sitting in while I'm recording from neurons in the spinal cord. I'm using a genetic, a transgenic line, and in this case that expresses the Voltron sensor in all the glutamatergic neurons in the spinal cord. So every excitatory neuron, or at least a large subset of the excitatory neurons in the spinal cord, express the sensor. And I record in a given focal plane, have the fish swim for a while, switch to the next plane, record more, and so step with a couple of optical sections through the spinal cord, and record the activity of as many neurons as possible. And this is sort of an example image here of the GFP tag. So because the Voltron sensor, as I mentioned, is very dim, it's actually very hard to see just by looking at it. So it has a green fluorescent tag on it. And this is the average image of a couple of tens of thousands of frames that are recorded during a few second imaging session. And then these are just a couple of example traces. And you can, and on the very bottom, you have the fictive swimming recording, the electrical readout that basically tells me when the fish is trying to swim. And you can see when it's swimming, there's some activity in some neurons and they look a bit different, and then it's not swimming anymore and most of the neurons become silent, and there's some diversity going on. And so the first thing that I wanted to do is basically characterize a bit this diversity that I'm seeing. And what I'm doing here is to use the fictive electrical recording that basically tells me when the fish is trying to swim and tells me the intended oscillation of the tail, and use this as a trigger to calculate the average voltage change over any swim cycle. So I'm using the VNR signal to trigger at every cycle and average the fluorescent traces for every neuron over one swim cycle, basically. And a neuron that is part of the central pattern generator that generates oscillation in the tail, on average, should have a strong modulation over an average swim cycle. Whereas a neuron that is active but doesn't take part in the oscillation itself, on average should sort of be a flat line. And this is shown in two example traces here. An oscillatory neuron and another neuron that is active. You can see the spikes here, but is not locked to the face of the oscillation itself. And this is the histogram of the sort of the modulation measuring over the whole data set. And you can see there's sort of two distributions, one sort of broader one that is of neurons that are modulated, and then there are neurons that have basically show no modulation during a swim cycle at all. And on the right here, so this is a sort of stylized transverse section of a spinal cord, and I'm plotting the position of every neuron in the coordinate system of a spinal cord. This would be where the axon tracks are, and then this is the central canal. And the color code shows how much the neuron is modulating its membrane potential during an oscillation. And you can see that there's a bunch of neurons over here in the sort of middle spinal cord that are highly modulated. And then there are other neurons, and mainly a big group of neurons over here that are not modulated and within one oscillatory cycle.
So actually, Urs, can you be a little bit like, with this kind of analysis, can you try to pinpoint to what would be, for instance, the V2A population that should be recruited for a given frequency? And do you know for sure that your ventral one are the V3?
So yes and no. I'm not modulating I'm not inducing behavior that modulates their swim frequency a lot. There's a bit of variation, obviously, but it also, yeah, all the oscillatory cells sort of fall in this region, and I don't, I can't be sure that these are all V2A neurons. I assume most of them are, but
you probably have other ones, right? But I'm just curious, like you should see so do you see the V0Vs that are very lateral and ventral? They're not coming up here, but they should be in the V too.
Yeah, it doesn't, lateral and ventral. No, I don't seem to see them a lot, no.
But you didn't try to play with the frequency of the gradient to see if you recruit different population?
Yeah, so if you look at any individual recording, there's probably some where you can see them, but putting the whole data set together, this is mostly what pops out. And I didn't try a lot of different conditions at any given imaging area, because as I mentioned, you don't have that much time before you bleach out your sensors. So I'll come back to that later, but I tried different grading speeds, but the swim frequency of the fish doesn't change drastically. So I think it's mostly in the sort of 20-25 hertz range what they're swimming here, and so I see the majority is the V2A neurons that are active here.
I have a question in line with what Claire just asked. I was wondering, so do you know the type of neuron? I didn't quite get it. Can you, like, from this data directly identify what type of neuron it is?
So the transgenic that drives the expression of the sensor drives it in excitatory neurons. So that I know, these are all excitatory neurons. More than that, I don't immediately know what type of cell I'm looking at, other than seeing a certain type of behavior and then going back to the literature and saying, okay, this neuron behaves like that. We know a neuron that behaves the spinal cord, so I assume it's, in this case, the oscillatory ones are probably are called V2A neurons because they are the main excitatory component of this of the central pattern generator in the larval zebrafish. But that's sort of by comparison to what's already known about their activity.
Okay.
I just wanted to show a bit more about the kind of sort of analysis that you can run if you have that kind of data. In this case, I just in the slide before, I just looked at the overall amplitude of modulation, but you can also look at the phase when in the oscillatory cycle a neuron is active and inactive. And if you do that, not surprisingly, you see two populations, one on the left side and one on the right side of the spinal cord. So in this example, in A here, we're looking at the top of a spinal cord, and the color code depicts phase within the, or the maximum amplitude within the oscillatory cycle. And there's basically a bunch of brownish cells on one side and a bunch of bluish cells on the other side. And two example traces here where you can see one is 180 degrees out of phase from the other one. And again, looking at only the cells that have a high, that are basically oscillatory and color coding by phase here, you can immediately identify two populations, and that is expected. There's one on the left side, on the right side, that oscillate basically back and forth. But what you can also do is look along the spinal cord, and now plotted here is on the y-axis the phase again, and on the x-axis the distance along the spinal cord. And you see the two populations, but you see they also have a slope, and that is because there's a slight phase shift from the head to the tail, because the excitation races down the tail. It starts at the head, races down the tail on one side, and then jumps basically back to the head and races down again on the other side. If you collapse the the two populations into one, and you can fit a slope here, and you get sort of the speed of the excitatory wave that races down the spinal cord that calculates to roughly half pi, so a quarter period per millimeter, which makes sense if you estimate that the tail is roughly 2 or so millimeters long, a bit longer maybe. You get half a period on one side, and then the other half of period on the other side of the tail. And these are the kind of measurements that you can do if you have this high speed recording of membrane potential, basically. And there are some cells that don't really sit on this line, and part of it is probably measurement noise, but part of it is also neurons that don't oscillate at the phase that you would expect them to be, given their position in the spinal cord. And you can start speculating that some of them might be other types of neurons that are slightly phase shifted. They could, for example, be MCOT neurons, some of them here, because they're expected to be somewhat phase shifted compared to V2A neurons. But I didn't dive into this in more detail. Okay, so now what I got really excited about was in the end, the other neurons that are not oscillating. The oscillatory ones are, it's really nice to be able to see them and measure all these things about them, but we sort of expected them to be there. The other neurons, it's much less clear what they represent. And this is again a sort of a plot now where I'm only plotting the ones that are not oscillatory, and the color code shows whether they are mainly active during swimming or outside of swimming. So you might know zebrafish larvae, they swim in bouts of activity and quiescence, and red indicates cells that are very active during swimming, and blue indicates cells that are active during quiescent periods. And there's just some sample traces of a neuron that is active during and outside of the swim episodes. So the black trace here again is the electrical fictive swimming recording. And this neuron is quite active, but as soon as the fish starts swimming, it actually falls silent and becomes very active again. And here is a neuron that is active during swimming. Whenever the fish is swimming, it starts spiking. But again, if you zoom in, the spikes are not phase locked to the actual oscillation of the swimming itself. I didn't dive into much of the analysis of these neurons that are active outside of swimming, just they're relatively sparse in my data set. You could hypothesize that they're, for example, part of a sensory system that gets shut off during swimming, so not to trigger any behavior that is due to self-motion or something like that. That's just speculation, but that would sort of fit with the observation that they're shut off during swimming.
So they are excitatory again, it's all bigger too?
Say again?
Oh, no, just all of them are excitatory, right? Still in this data, right? We're looking at
yes, it's still the same. Yeah, okay. It's all excitatory neurons.
And is there any correlation with the visual stimulus? Like if you stop the visual stimulus, if you do the same experiment and look at spontaneous swimming, would you see the same or not? With calcium imaging,
yeah, I sort of expect to see the same, but I did not do that experiment. I didn't do that experiment. Yeah. My hypothesis would be that it's uncorrelated of any visual stimulus. The visual stimulus I'm in this case more using to make the fish swim in the first part and sort of have it swim, yeah, at a high enough rate so I can actually get data in a relatively short amount of time. But yeah, I didn't compare that very carefully. so what I got interested in is basically this ventral population, very ventral excitatory population of neurons that are active during swimming, but that are not, seemingly not part of the central pattern generator because they're not actually oscillating. And this is just to show that again, basically their spike probability within one oscillation is basically flat. They spike at any point during the oscillation, and this shows the ratio of whether they mainly of spikes during swimming and outside of swimming. And basically what it shows is that it's shifted towards the right. So most of the spikes occur when the fish is swimming, and when the fish is not swimming, they're mainly silent, although it's not as clear cut. And okay, so just wanted to show you some sort of side note of another sort of type of analysis that you can now run with this kind of data. This is a field of view of these ventral neurons, where you see it's an average, the average of the recording, and it's quite messy. It's hard to see much. You can sort of make out the cell bodies, and these are the respective traces, and they'll start firing, and then they're silent again, and so on and so forth. But on the at high time resolutions, their spiking is actually quite uncorrelated. So if you now pick any cell and generate a spike triggered movie, meaning at every spike you take a little bit of the recording before and after the spike and average over every spike the cell is doing, since the other neurons are spiking in an uncorrelated fashion, you basically end up with a movie that only shows this one single neuron. And I'm not going to play the movie again because it's going to be hard, but if you then use the average spike and generate a correlation map of how well correlated every pixel in that movie is with the average spiking of that neuron, you end up with something like that. And basically what you can see now is suddenly the local morphology of the neuron, of the cell body again, and then some of the presumably dendrites or maybe axon, hard to say, that is visible, that comes out in a correlation analysis like that, because on very short time scales, their spiking is actually uncorrelated of each other. And you could do that for every neuron, color code it, and then create a nice map like that, where every individual neuron now pops up with its sort of local morphology, as compared to just the average image of the fluorescent signal, where you can't really recognize anything. Okay, so quick conclusion.
Hi, I have a question for the previous slide. Yeah, how do you need a long recording to get this?
Yeah, I mean, it depends. You don't need necessarily a super long recording, but you need sufficient spikes in that recording, right? So this recording, I think it was only maybe 30 seconds long, but there are maybe 100, 200 spikes or more in there. So, so yeah, I think that's sort of what you need to get a nice measurement like that. And obviously they need to be sufficiently uncorrelated to separate the individual neurons.
And what is the FPS for the camera to use?
So all my recordings are done at 1 kilohertz. Yeah.
Okay, thanks.
Okay, so in this first part, I showed you that I'm using voltage imaging in excitatory neurons in the spinal cord to get an overview of the diversity of activity that exists during a normal swim. And you can roughly separate them in oscillating and non-oscillating cells. And the oscillating neurons are probably mainly what are called V2A neurons, which make up the main excitatory component of the larval zebrafish, at least at these swimming frequencies. And there's a large group of non-oscillating neurons that are situated at the very ventral spinal cord that are active during swimming, but they're not oscillating.
Can you say that those are V3, or can you check whether they are V3? Because you know the V3 are extremely hard to recover from with electrophysiology. Like me going in the lab had some recording of the cells. It's extremely hard to reach because they're in a location where you need to go through the axons.
Yeah,
so we have some recordings of them.
I have some data why I think they're V3 neurons. I'm coming to that. So okay. I think they're V3 neurons because so that's what I hypothesized. I wanted to sort of get a bit of a better idea whether that's true. On the right, there's a schematic of basically the glutamatergic interneurons at the the age where I'm using the fish at, and I use single cell photoconversion. So all the neurons express a protein called K80 that can switch from green to red upon UV stimulation. I'm parking my laser on one single neuron that converts the protein from green to red. Wait a day for the protein to diffuse into the whole neuron. Come back and image it and look at their morphology. And basically, what I can see is that they have like a relatively local, what I think is a dendritic arbor, and then they have a super long axon that goes, in some cases, basically the entire tail of the larva, and that is in accordance with what has been previously described for VME on the scheme, but V3 neurons. I think there are actually two morphological types. So I saw the ones that have this very long descending axon. I also saw some that have a bifurcating axon that goes ascending and descending. So so these are not really described so far. And it's unclear whether they are just two morphological types of the same sort of neurons or whether these are different neurons that have not been described before.
Subset of those are serotonergic, right? Like when we've done the immunostaining for serotonin, we've seen that there are a couple of serotonergic neurons in there.
Yeah. Yeah, I didn't distinguish them. I didn't do any staining for serotonin at this point. I also don't know, I can't tell for sure whether functionally I see both of them doing the same thing or whether I'm mainly looking at one or the other type, because not all the neurons are always active in that area, and I currently don't have a good way to say whether the neuron that was active, is it the one that has a descending axon or a bifurcating axon. But I think basically. So the question then, what I wanted to get a bit into at least, is what these guys are doing. What's their sort of role in the circuit? And the first clue came from changing the virtual environment of these that the fish are swimming in. So I told you that I'm basically, the fish gets to its own spinal cord activity, and there's basically one knob that you can play with, is the gain of this feedback. So for a given amount of swimming activity, the grading that the fish sees is either moves a lot or a little. And if it moves a little, the fish tries to swim harder because it wants to maintain its position in the, it's this virtual flow. And this is basically shown here. When the gain of this feedback drops, there's more stronger activity. And if I record from these neurons at the same time, sometimes you can see a dramatic change in their activity. So under high gain situation where the fish swims less strongly, they're hardly active at all. And then once the fish wants to swim more strongly because it wants to keep, it swims in a low gain scenario and wants to keep up with the optic flow, suddenly become very active. And so over the whole data set, the data is a bit more noisy, but I think there's a sort of a correlation between how strong the fish swims, sort of the power in any given bout, and how and the spike rate of these neurons, basically. And maybe it's not actually a linear relationship, but more sort of a sigmoidal one. It's not exactly clear, but I think what these neurons are, they're mainly active when the fish wants to swim more strongly. And so the last sort of experimental slide that I have is basically trying to get a bit more at the, putting, so far this is sort of more a correlation that I'm seeing, and I want to see whether they can actually modulate how strong a fish swims. So my hypothesis now would be that these neurons modulate something about the strength or the amplitude or power of the fish as swimming. And to do that, I'm moving away from voltage imaging, and we were lucky to get our hands on a combination of transgenic lines that express general rhodopsin specifically in V3 neurons. And so basically what that allows me is to activate these guys specifically. And I'm also going away a bit from this in vivo as natural as possible kind of swimming, but to get rid of any feedback effects from the brain or visual responses to any general rhodopsin activation. I'm actually spinalizing the fish. So I basically cut the spinal cord after the hindbrain. And usually fish wouldn't do anything in that kind of situation. They're not swimming at all. But what you can do is you add NMDA to the solution, and that just gives a tonic excitatory drive to the entire spinal networks. And at a sufficiently high enough drive, they start wiggling their tail again. Basically, this is an effect that's been known for in many systems for a long time. But basically, you can have an oscillating spinal cord that is cut off of any sort of modulatory feedback from the brain or influence from the brain. And in these kind of fish, I'm then comparing how the swimming looks like when I additionally stimulate the V3 neurons. And it looks like that. So these are basically just average swimming over many trials at different NMDA concentrations. In the first 3 seconds, the light is on. In the later half of the trace, the light is off. And what you can basically see is that the fish swims more strongly in when I activate these 3 neurons. And this is sort of lumped together.
What is the y-axis here?
The y-axis is just, so this is a low-pass filtered rectified tail angle. So it's a bit an arbitrary axis. So this increase here could be to higher amplitude swimming, could also be more swimming in general. So on average, it's higher. If you look at, so the effect is sort of most visible in that plot. But if you look at individual swims, amplitude or duration or frequency, you sort of see a similar effect, which is that here on the y-axis, I basically have the amplitude of any given swim when the light is on, and on the x-axis when the light is off. So if activating V3 neurons with light has an effect on the swimming, all the dots should be above the line, basically. And this is, I think, what I'm mainly seeing is that the amplitude increases when V3 neurons are activated. The duration also seems to increase in most of the cases. And each dot here is the average of several swim episodes in one fish. And sort of importantly, I think the frequency does not change a lot. If it changes very little. And this is interesting, I think, because most of the spinal networks in zebrafish so far have been looking at what type of neurons become active at different frequencies. At higher frequencies, different neurons get recruited, different motor neurons get recruited, and these changes. Whereas this, I think, is more a system that within a given sort of frequency range can modulate the strength of the swim output, either by having the bouts being longer or the tail oscillate with a higher amplitude. Okay, this is sort of like moving away from the imaging and trying to understand more about the actual functional role of the neurons that we sort of identified with our voltage imaging approaches.
Just a little, like few remarks here. I think knowing that the subset is serotonergic, it would be neat to know how much this effect you're seeing, it could be serotonergic dependent. If you block it with, or you reduce it with blockers.
Yes.
And then the second thing is that based on their morphology, it's kind of a question of that they can't project, like at least the one that you showed us, because there are multiple kinds, but they look quite ventral in their organization as well, right? So
they look relatively, yes. You mean in terms of like projecting onto motor neurons?
If they project onto the ventral motor neurons, they won't change the power of swimming, right? Like the amplitude won't change because they need to reach dorsal motor neurons to do that. So what they could, it's really a matter of looking carefully at your morphology, but it could be that they project onto the inhibitory and GRIL, for instance, or the V2B, the V1.
Why do you say they wouldn't be able? Why would they not be able to change amplitude?
Power is like related to the motor neuron class that you're targeting. So if they're really, really ventral and they're going for the small secondary that project onto the slow muscle fiber, it wouldn't increase the power of swimming. They would need to reach out to the intermediary secondary motor neuron, at least, right? But that's why I think the morphology of the cells is really telling you a lot about where they could potentially, what they could reach in terms of motor neurons or inhibitory interneurons. No?
Yeah, it's a good point.
It's nice to look at this carefully, I think. And it goes, what you're observing is quite, because we know very little about these cells in other species too. It's really interesting to dig into it.
Yeah, it's a good point. We can talk about it later if you want. Okay, I just want to conclude. The second part is basically, I think that these neurons that are active during swimming are not oscillating our V3 neurons. They seem to have distinct morphology that basically innervate with their axon the entire tail. They seem to increase their activity when the larvae want to swim more strongly. And when I activate these neurons, that seems to have an effect on how strong the tail moves. So these two together, my current working hypothesis is that these neurons are sort of a module that lets you tune the sort of output of the tail in a given swimming regime, which in this case would be slow swimming, so forward swimming. And that these neurons are sort of a knob that the network can use to modulate the power of any given swim. Okay, with that, I'm going to just acknowledge the people that I've been working with. As mentioned, I'm working in Adam Cohen's lab, and we're doing a lot of voltage imaging, and everybody's been very helpful. The genetic lines to do the channel dopsin came from Shinichi Higashishima in Japan, and Boyden and Arns' lab, and have been providing sensors and fish lines. And then I'm also collaborating with Ryan Engert across the street for some of the projects. Thank you for listening to this and being here.